Hello friends, welcome to Unlocked Science by Mishtab. No criming, just understanding. Have you ever wondered what makes complex life complex? Meet the eukaryotic cell, the ultimate biological upgrade. By end of this video, therefore, you'll be able to define a eukaryotic cell and give examples of eukaryotes, identify key features of eukaryotic cell, differentiate eukaryotic cells from prokaryotic cells, explain the importance of cells in medicine. To achieve all those outcomes, we first need a clear definition. So, what is a eukaryotic cell? A eukaryotic cell or eukaryotic cells are cells which have distinct nucleus and their DNA material is bonded by a membrane. Eukaryotic comes from two words, eu means true and karyo means nucleus. Therefore, eukaryotic means true nucleus, accounting for distinct nucleus in the definition. These cells are said to have evolved from prokaryotic cells, and organisms containing eukaryotic cells are called eukaryotes. Examples we have plants, animals, and fungi. Those are some of the examples of eukaryotes, but what exactly makes up a eukaryotic cell? Let's dive into the major parts. The first part is the cell membrane. The cell membrane, sometimes called plasma membrane, is the outer surface of the cell that separates the internal contents from the external environment and controls movement of materials in and out of the cell. Structurally, the cell membrane is made up of phospholipid bilayers whose surface contain protein molecules floating about them to a fluid mosaic pattern, hence the name fluid mosaic model. Each phospholipid molecule has a phosphate head which is hydrophilic and therefore water-loving, and two lipid tails which are hydrophobic and therefore water-hating. The hydrophobic tails face inwards, away from the water, while the hydrophilic heads face outwards towards the watery environments, inside and outside of the cell. This arrangement forms a stable barrier between the inside and outside of the cell. Within the phospholipid bilayer, there are proteins. Some are floating, some on the surface, and others span through. Proteins that span through the phospholipid bilayer are called intrinsic proteins or integral proteins, and these act as transporters, carriers, allowing materials like ions, nutrients to pass through the membrane. Proteins on the surface of the cell membrane are called peripheral proteins or extrinsic proteins, and these act as enzymes, receptor sites. Some extrinsic proteins are also attached to a carbohydrate group and hence called glycoproteins. Some extrinsic proteins are also attached to cytoskeletons. Some extrinsic proteins maintain the cell shape by linking the cytoskeleton inside the cell. The membrane also contains cholesterol that distort the closed packaging of phospholipids to keep them more fluid. Cholesterol also increases the stability and flexibility of the membrane. Therefore, the membrane cannot break out in its presence. The cell membrane also contains carbohydrates that are attached to lipids or proteins. Those attached to the lipids are called glycolipids, and those attached to proteins are called glycoproteins. Functions of the cell membrane. Protection and support. The cell membrane acts as a barrier between the internal environment of the cell and the external surrounding. It maintains the cell shape and protects the internal organelles from damage. Selective permeability or control of transport. The cell membrane is selectively permeable to regulate the entry and exit of substances, allowing essential substances such as oxygen, water, and nutrients to enter, and lets out waste products like carbon dioxide, which maintains homeostasis, that's the balance of the inside environment. Communication and signaling. The membrane contains receptor proteins that help the cell receive signals like hormones or neurotransmitters from other cells. This allows the cell to respond to its environment, for example, reacting to changes in temperature or presence of chemicals. Cell recognition and identification. The outer surface of the cell has glycoproteins and glycolipids, that's proteins and lipids with carbohydrates, that act as ID tags, helping cells recognize each other, especially in tissue formation, immunity, and organ transplantation, flexibility and growth. The membrane's fluid nature also allows the cell to change shape, grow, and divide. It also enables endocytosis, that's taking in of materials, and exocytosis, that's taking out of materials. Attachment and interaction. The cell membrane provides anchoring sites for cytoskeleton inside the cell and extracellular matrix molecules outside. This strengthens the cell and helps it connect to other neighboring cells. The second part is cytoplasm. Cytoplasm is a gel-like substance inside the cell that fills the space between the cell membrane and the nucleus. It contains the cell internal contents except the nucleus. It comprises cytosol, cytoskeleton, and organelles. Cytosol is the liquid portion of cytoplasm containing 80% water and dissolved mineral salts, proteins, and small organic molecules like glucose and amino acids. In prokaryotes, it contains the DNA. Cytoskeleton. Cytoplasm also contains a network of protein fibers called cytoskeleton that gives the cell shape and helps organelles move around the cell. It contains small specialized structures like mitochondria, ribosomes, Golgi apparatus, and endoplasmic reticulum. Functions of the cytoplasm. It's a medium for most of the chemical reactions like glycolysis, and many metabolic pathways occur in the cytosol. It suspends most of the organelles and holds them in position. It moves organelles around the cell and allows movement of materials inside the cell by cytoplasmic streaming. Storage. It stores nutrients like glucose, amino acids, wastes, and minerals. The cytoplasm also gives the cell shape and support. Homeostasis. It maintains the internal environment constant and balances pH and water. The next part is nucleus. It's the control center of a eukaryotic cell. It's a membrane-bound organelle that contains the cell's genetic material, that's deoxyribonucleic acid DNA, that's wrapped around proteins forming chromatins, and when the cell is ready to divide, chromatins coil more tightly forming chromosomes so that the DNA can separate easily. It's enclosed by a double-layered membrane called nuclear membrane that is perforated by the nuclear pores to regulate the transport of molecules between the cytoplasm and the nucleus. It contains dense central structure called the nucleolus and also contains ribosomes that are manufactured by the nucleolus. Functions of nucleus. Genetic material storage. It houses the cell's genome, which is organized into multiple linear chromosomes. Controls gene expression. The nucleus regulates which genes are turned on or off by controlling access to DNA and the process of transcription and RNA processing. DNA replication and transcription, that's copying of DNA to RNA, occur within the nucleus. Ribosome biogenesis. The nucleus is also a site for ribosome assembly, which are essential for proteins. Control cell activities. It regulates metabolism, growth, and reproduction. Next is mitochondria. It's the powerhouse of a eukaryotic cell. Mitochondria is a double membrane bond organelle responsible for generating the cell's energy in the form of ATP, that's adenosine triphosphate. The double membrane has the outer membrane and the inner membrane. The inner membrane is highly folded inwards, giving rise to extensions called cristae. The region between the two membranes is called the intermembrane space. The inner membrane encloses a fluid called mitochondrial matrix that contains enzymes for Krebs cycle, DNA, and ribosomes. Mitochondria also contains a small circular genome within the matrix that encodes some of the proteins needed for mitochondrial function, and therefore it is said to be having its own DNA. Mitochondria have their own ribosomes, which are used to synthesize proteins from the instructions of the mitochondrial DNA. Functions of the mitochondria. Energy production. The primary function of the mitochondria is to break down glucose during cellular respiration to release energy. Regulate metabolism. Mitochondria help control metabolic activities. It stores calcium ions important for cell signaling. In specialized brown fat cells, mitochondria produces heat during metabolism, and this helps maintain the internal environment constant. Apoptosis. The mitochondria help control cell death. Apoptosis is controlled cell death, and when needed, the mitochondria helps control it. They act as cell guardians, in that when the cell is damaged or dangerous, mitochondria triggers cell death by releasing cytochrome C into the cytoplasm, which activates enzymes called capazanes that destroy the cell in a clean and controlled way. So those are the functions of the mitochondria. And then there is a unique origin of mitochondria that drives us to endosymbiotic theory. Mitochondria originated from free-living bacteria that were engulfed by early cells and formed a mutually beneficial relationship. This explains why mitochondria have their own DNA, which is circular, double membranes, and replicate like bacteria. However, mitochondria are semi-autonomous organelles because they replicate independently. They have their own DNA and make some of their own proteins, but they still depend on nucleus and cytoplasm for many essential proteins. They cannot live outside the cell, so they are not fully autonomous. Golgi apparatus is the fifth part. It's a membrane bond organelle responsible for modifying, packaging, and transporting proteins and lipids inside the cell. Golgi is made up of a stack of flattened disc-shaped sacs called cestani, typically numbering from 4 to 8, though this can vary. It has a distinct polarity, with the receiving cis face, which is convex, and that faces the endoplasmic reticulum, and a shipping transface, which is concave and faces away from the endoplasmic reticulum. So the cis part is for receiving and trans part is for exiting, or where the vesicles exit from. The Golgi is connected to the rest of the cell through a network of vesicles. Functions of the Golgi apparatus. Modification. Golgi apparatus further processes proteins and lipids received from the endoplasmic reticulum, often by adding or modifying a carbohydrate chain. It also sorts the molecules, tagging them for further destinations. The Golgi apparatus packages the processed molecules into vesicles for delivery into other cellular locations such as lysosomes, the plasma membrane, or for secretion outside the cell. Carbohydrate synthesis. In the plant cells, the Golgi also synthesizes complex carbohydrates needed for the cell wall. Dive further into the endoplasmic reticulum. Endoplasmic reticulum is a large network of membrane bond tubes and sacs in the cell that plays a major role in protein and lipid synthesis, transport, and detoxification. It is connected to the nucleus and helps move materials throughout the cell. It's like the highway inside the cell. It is divided into two types, that's the rough endoplasmic reticulum and smooth endoplasmic reticulum. The endoplasmic membrane is continuous with the outer nuclear membrane extending throughout the cytoplasm. Rough endoplasmic reticulum is characterized by the presence of ribosomes on its surface, which gives it a rough appearance. Smooth endoplasmic reticulum is a network of tubules that lack ribosomes on its surface. Functions of endoplasmic reticulum. Protein synthesis and modification. The rough endoplasmic reticulum synthesizes, folds, and modifies proteins. Lipid synthesis. The smooth endoplasmic reticulum is the primary site for synthesis of lipids, steroids, and phospholipids. Transport. The endoplasmic reticulum acts as a transport system, moving proteins, lipids, and other molecules to different parts of the cell. Calcium storage. The endoplasmic reticulum serves as the major storage of calcium ions, which are crucial for cell signaling and muscle contraction. The smooth endoplasmic reticulum can detoxify harmful substances in liver cells, and therefore liver cells contain abundance of smooth endoplasmic reticulum. Ribosomes. Ribosomes are molecular machines in all cells responsible for protein synthesis by translating genetic information from messenger RNA to amino acids. A ribosome is a small, dense, and non-membrane bond organelle comprising ribosomal RNA and proteins. Each ribosome has two main parts, a small subunit that reads the messenger RNA and a large subunit that links the amino acid together. Functions of ribosomes. Protein synthesis. Ribosomes read the sequence of codons on messenger RNA, that is mRNA, molecule, and use this information to assemble a polypeptide chain of amino acids. Translation. This is the process of building a protein from messenger RNA, and it is done by a ribosome. Polypeptide chain formation. The small subunit binds to the messenger RNA, while the large subunit catalyzes the formation of the peptide bonds between amino acids, which are derived by transfer of RNA molecules. Protein release. Once the chain is complete, it is released from the ribosome and folds into a functional protein. Ribosomes are found in the cytoplasm of both prokaryotic and eukaryotic cells. Prokaryotic cells contain 78 ribosomes and eukaryotic cells contain 88 ribosomes. There are free ribosomes which are found in the cytosol, and these produce proteins that will function within the cell itself. There are bound ribosomes which are attached to the endoplasmic reticulum, and these produce proteins that will be secreted or used in membranes. Mitochondrial and chloroplast ribosomes, and these are found in the mitochondria and chloroplast respectively. So we have three types of ribosomes. That's the free ribosomes, bound ribosomes, and mitochondrial or chloroplast ribosomes. Then our next part is peroxisomes. A peroxisome is a single membrane bond organelle that contains a granular matrix filled with enzymes. These ribosomes are responsible for carrying out a variety of metabolic reactions, including breakdown of fatty acids, amino acids, and detoxification. They are found in almost all eukaryotic cells. Functions. Fatty acid oxidation. Peroxisomes break down fatty acids for metabolic energy. Detoxification. Their matrix contains catalase enzyme that catalyze the breakdown of toxic hydrogen peroxide produced during oxidation reactions into water and oxygen. Biosynthesis. Peroxisomes are involved in the synthesis of specialized phospholipids, such as those needed for nerve cell myelination. Other processes, like synthesis of cholesterol. They play a big role in the synthesis of cholesterol and bile acids and photorespiration in plants. Origin and replication. Peroxisomes lack DNA. Unlike mitochondria and chromosomes, they do not contain their own DNA or ribosomes, and they import all their enzymes from the proteins that are synthesized on free ribosomes in the cytosol. Peroxisomes replicate by growing and then dividing, or through a process involving other organelles like the endoplasmic reticulum. They are found in liver cells and kidney cells to carry out detoxification and waste metabolism, and in brain cells to carry out synthesis of the nerve cells. Lysosomes. This is the cell's digestive system with a single lipid bilayer that keeps the powerful digestive enzymes and prevents them from damaging the rest of the cell. The inside environment is acidic, that's its pH is 5, because of the protein pump in the membrane that actively pump hydrogen ions into the organelle. It has a varied appearance in size and shape, depending on the material they are digesting. Functions of the lysosomes. Intracellular digestion. They contain digestive enzymes that catalyze the breakdown of carbohydrates, proteins, lipids, and nucleic acids. They digest materials brought into the cell by endocytosis, phagocytosis, and pinocytosis. Autophagy, that is self-eating or recycling. Lysosomes break down old, worn-out organelles and recycle their components, but the cell renews itself by repressing damaged structures. Autolysis, which is cell suicide. Uncontrolled cell death where lysosome bursts and releases all the enzymes inside the cell, causing complete digestion of the cell. This occurs when the cell is severely damaged, aged or non-functional, or infected or toxic. Apoptosis. This is a controlled and planned cell death where lysosomes release some enzymes into the cytoplasm And these enzymes break down cellular components, work together with the mitochondria, and ensure killing and cell death. Embryonic development. During embryonic development, lysosomes help shape the body by breaking down and removing cells that are no longer needed. They participate in programmed cell death and tissue remodeling, allowing structures like finger and organs to form correctly. Defense against pathogens. Lysosomes destroy bacteria, viruses, and foreign particles that enter the cell, playing a vital role in immune cells like macrophages and neutrophils. The next part is the centrosome. A centrosome is a non-membrane bond organelle located near the nucleus of most animal cells. Therefore, the centrosome is only found in animal cells. It acts as the main microtube organizing center and plays a crucial role in cell division, cell shape, and movement of organelles. It is sometimes called the cell's organizing headquarters for the cytoskeleton. It comprises two cylindrical structures called centrioles, arranged perpendicular to each other. Each centriole has a 9 by 3 microtubule arrangement, that's 9 triplets or microtubules. It contains a protein-rich matrix surrounding the centrioles called the pericentriolar material, where microtubules grow from. Therefore, centrioles plus pericentriolar material gives us a centrosome. Functions of the centrosome. Microtubule organization. It acts as the main microtubule organizing center in animal cells, nucleating and organizing microtubules that form the cytoskeleton. Cell division. During cell division, the centrosome duplicates. The two centrosomes move to the opposite poles of the cell and organize the mitotic spindle, which separates the chromosome into two daughter cells. Cell polarity. Centrosomes play a role in establishing cell polarity and can position the nucleus within the cell. Cilia and flagella formation. They are also responsible for assembly of cilia and flagella by docking at the cell membrane. Well, these centrosomes vary in eukaryotes. Animal cells contain a centrosome with two centrioles embedded in the pericentriolus material, and other karyotes like fungi and amoeba lack centrioles. They have their structures such as spindle pole bodies or nucleus-associated bodies that serve the same function as that of the centrosome. Next is cytoskeleton. This is a dynamic network of protein fibers found throughout the cytoplasm of eukaryotic cells. It acts as the cell's internal framework or skeleton, giving the cell shape, strength, and support, and enabling movement inside and outside the cell. It is composed of three main types of filaments: microtubules, which are the largest, intermediate filaments, which are mid-sized, and actin filaments or microfilaments, which are the smallest. Microtubules are large hollow tubes made up of protein tubulin. They are crucial for intercellular transport, cell division, that's forming of the spindle fibers, and formation of cilia and flagella. Intermediate filaments. They also contain the intermediate filaments that provide mechanical strength and structure support to the cell. Their specific protein structure varies depending on the cell type. The actin filaments are the smallest and are made up of protein actin. They are involved in cell shape, changes, muscle contraction, cell motility, and cytoplasmic streaming. Functions of cytoskeleton: structure, support, and shape. Cytoskeleton provides a rigid framework that gives the cell shape and prevent it from collapsing. Organelle organization. Cytoskeleton anchor organelles in specific locations, allowing them to function and interact properly. Intracellular transport. Cytoskeleton act as highway system with motor proteins, moving vesicles, organelles, and other materials around the filaments. Cellular movement. Cytoskeleton enables cell motility and movement, including the contraction needed for movement and formation of structures like cilia and flagella. Cell division. They play a vital role in separating chromosomes during mitosis and meiosis through action of the microtubule, forming the spindle fibers. Signal transduction. They help integrate signals from the cell's movement, influencing cellular behavior and communication. Our next part is vacuole. It's a membrane organelle found in cells that are used for storage, support, and maintaining internal balance. In plant cells, it's a large central vacuole, which is crucial for maintaining the cell shape and rigidity through the tug-up pressure. In contrast, in animal cells, they have small numerous vacuoles for waste storage and other functions. Functions of the vacuoles. Storage. Vacuoles store water, inorganic ions, storage and proteins. They can also store pigments and defense molecules like toxins. Waste disposal. They serve as cellular garbage dump for waste products and harmful toxins, which are later broken or expelled. Tug-up pressure. In plants, the central vacuole maintains tug-up pressure, which is the force of water pushing against the soil wall. This pressure keeps the plant rigid and upright. Digestion. Plant vacuoles contain hydrolytic enzymes and act like lysosomes, breaking down large vacuoles. Cell growth. The vacuole contributes to the plant cell growth by expanding and absorbing water, pushing cytoplasm against the cell wall. Plants typically contain large cell vacuole that is centrally located and is enclosed by a membrane called tonoplast that has transporters to regulate passage of materials. Then in animal cells, they have small numerous vacuoles that are often dynamic. Chloroplast. It's a double membrane bond organelle found in plant cells and some algae responsible for photosynthesis. The double membrane is also called chloroplast envelope, and this regulates passage of molecules between the stroma and cytosol. Inside the envelope is a third internal membrane system that forms flattened sacs called thylakoids. These are sites for light-dependent reactions of photosynthesis. Thylakoids are often stacked in two columns called grana, which are interconnected by a network of membranes. The fluid-filled space surrounding the grana is called the stroma. It contains enzymes, DNA, ribosomes, and starch grains, and is a site for light-dependent reactions. That's the Calvin cycle. Functions. Photosynthesis. The primary function is photosynthesis, where light energy, carbon dioxide, and water are converted into glucose and oxygen. Energy conversion. Chlorophyll and other pigments within the thylakoid membrane absorb light energy to create ATP, which is an energy-carrying compound. Carbohydrate synthesis. The ATP from light reactions are also used in the stroma to convert carbon dioxide into sugars during Calvin cycle. Chloroplasts are also involved in biosynthesis of fatty acids, amino acids, and other organic compounds essential for the cell. Cell wall. This is a strong rigid outer layer found outside the cell membrane in plants, fungi, bacteria, and some protists. In plants, it is made up of cellulose, which forms long chains, and microfibrils embedded in a matrix of hemicellulose and pectin. In fungi, it comprises chitin. In algae and protists, it is made up of various substances including silica, calcium, carbonate, depending on the organism. Functions of the cell wall. Structure, support, and shape. The cell wall provides rigidity and a defined shape of the cell. Protection. It acts as a protective barrier against injury, pathogens, and environmental stresses like desiccation. Osmotic protection. The cell wall prevents the cell from overexpanding where water enters and is vital in tug-up pressure. In animal cells, which lack cell wall, but they do not usually burst. Animal cells lack cell wall, but they do not usually burst because they contain the water balance through osmoregulation. The cell membrane, cytoskeleton, and controlled internal environment prevent excessive water entry. In the body, fluids are kept isotonic, preventing swelling and bursting. Filtering. It can act as a selective barrier, controlling what substances can pass through the cell membrane. Understanding the structure of the cell helps us understand how complex life can be. Next, let's compare prokaryotic and eukaryotic cells. See how they vary in structure and complexity. Both eukaryotes and prokaryotes possess some membrane, cytoplasm, and ribosomes. Then, differently, prokaryotes are smaller, eukaryotes are larger. DNA in prokaryotes is not enclosed in the nuclear membrane, Instead, it is found in the cytoplasm in a region called the nucleoid and it is circular. Whereas in eukaryotes, DNA is enclosed within a membrane-bound nucleus and is linear, arranged in chromosomes. Prokaryotes have 78 ribosomes, while eukaryotes have 88 ribosomes. Prokaryotic cells possess mesosome for respiration. Eukaryotic cells possess mitochondria for respiration. Prokaryotic cells lack membrane-bound organelles, while eukaryotic cells possess membrane-bound organelles. With a clear understanding about prokaryotic and eukaryotic cell, let us quickly discover how these cells are important in medicine. Understanding diseases. By studying cells, scientists and doctors understand how diseases start and progress. Many diseases like cancer, diabetes, and HIV begin at cellular level. For example, cancer happens when the cell grows and divides uncontrollably. Studying cells also help doctors diagnose and develop treatments. Drug treatment. New medicine are tested on cells to see how they respond. This helps scientists to discover how drugs affect cell functions and whether they are safe or effective before testing them on humans. Antibiotics target bacterial prokaryotic cells without harming human eukaryotic cells. Vaccines. Cells help in development of vaccines which train the body's cell, especially immune cells, to fight against diseases such as COVID-19 and polio. Organ transplant and tissue culture. Understanding how cells grow helps in culturing tissues in laboratory and making organ transplants safer through compatibility testing. So that's all about eukaryotic cells, the complex and well-organized building blocks of plants, animals, fungi, and protists. Understanding them helps us appreciate how living things function. Don't forget to like, share, subscribe for more science lessons. Bye-bye.